One of the coolest things about fungi is definitely the diverse shape and forms of mushrooms. But how does the filamentous cells end up becoming this giant organ? Welcome back to Fungal Friday. Today we are diving into how mushrooms are formed. Fungi usually grow in filamentous form called mycelia. When the conditions are right, they form mushrooms, their reproductive structures. Different species of fungi develop mushrooms in unique ways. But in the model mushroom, Coprinopsis cinerea, or inky cap, it starts with hyphal knots and develops into primordia. Inside the primordia, we can already see tiny mushrooms beginning to form. As they mature, the outer layers, called veils, break apart and become the shag covering the cap and stem. Although we have a grasp on the general developmental stages, the molecular mechanisms behind how fungal tissues differentiate are largely unknown still. Recently, Fuddy and collaborators investigated an interesting gene that is nearly completely inactive in vegetative mycelia. When they deleted this gene, the mushroom development was arrested at an early stage. The fungi stayed as these cute, fluffy primordia instead. Because of this, the gene was named Snowball1. And unlike the normal inky cap, the Snowball1 mutant does not have a tiny mushroom inside their primordia. This phenotype is similar to the mutant of another gene, Ichijiku1, or no flower fruit in Japanese. This gene encodes a nuclear methyltransferase and can remodel the chromatin, controlling global gene expression. So what does Snowball1 do? Is it also a methyltransferase? Actually, no. Based on its structure, Snowball1 is predicted to be a membrane protein with 7 transmembrane helices, similar to a G protein couple receptor, or GPCR. However, it lacks the typical sequence motifs found in the classical GPCRs, so its exact function is still unclear. To learn more, Fuddy's team turned to a transcriptomic approach to compare gene expression between normal and mutant fungi. They found that Snowball1 affected the expression of many genes involved in cell growth, cell wall formation, and tissue differentiation. And most intriguingly, Ichijiku1 was also significantly downregulated in the Snowball1 mutant. Based on my very limited knowledge of this topic, here's one possibility. During mushroom formation, some localized signals might activate Snowball1. Snowball1 further regulates the activity of Ichijiku1, which controls the expression of the downstream genes to coordinate mushroom development. Lastly, Snowball1 is found in other fungi as well, and it is often upregulated during mushroom formation. Whether it is essential across species is still an open, fascinating question. And maybe following genes like Snowball1, we will finally know how mushrooms are made. With that, thank you for joining me today, and I hope to see you next week.